Borrelia burgdorferi, Wikipedia article audio. Borrelia burgdorferi is a bacterial species of the spirochete class of the genus Borrelia. B. burgdorferi exists in North America and Europe and is the only causative agent of Lyme disease in the United States. Borrelia species are considered diderm bacteria rather than gram-positive or negative. Borrelia burgdorferi is named after the researcher Willy Burgdorfer, who first isolated the bacterium in 1982. Borrelia species in the species complex known to cause Lyme disease are collectively called Borrelia burgdorferi sensolato, not to be confused with this single species Borrelia burgdorferi sensu stricto in that complex which is responsible for all cases of Lyme disease in North America. Microbiology. Morphology. B. burgdorferi resembles other spirochetes in that it has an outer membrane and inner membrane with a thin layer of peptidoglycan in between. However, the outer membrane lacks lipopolysaccharide. Its shape is a flat wave. It is about 0.3 μm wide and 5 to 20 μm in length. B. burgdorferi is a microaerobic, motile spirochete with 7 to 11 bundled perisplasmic flagella set at each end that allow the bacterium to move in low and high viscosity media alike, which is related to its high virulence factor. B. burgdorferi is a slow-growing microaerophilic spirochete with a doubling time of 24 to 48 hours. It is one of the few bacteria that can survive without iron, having replaced all of its iron-sulfur cluster enzymes with enzymes that use manganese, thus avoiding the problem many pathogenic bacteria face in acquiring iron. B. burgdorferi circulates between ixodes ticks and a vertebrate host in an enzootic cycle. B. burgdorferi living in a tick cannot be passed to its offspring. Therefore, tick larvae must feed on the blood of an infected animal to acquire B. burgdorferi, which ends up in the midgut. The spirochetes survive as the larvae molts into a nymph and persist in the nutrient-poor midgut as the nymph overwinters. Infected nymphs then transmit B. burgdorferi by feeding on another vertebrate to complete the cycle. Ticks can transmit B. burgdorferi to humans, but humans are dead-end hosts, unlikely to continue the life cycle of the spirochete. Nymphs molt into adult ticks, which usually feed on larger mammals that are not able to support the survival of B. burgdorferi. Lyme disease is a zoonotic, vector-borne disease transmitted by the ixodes tick. The infected nymphal tick transmits B. burgdorferi via its saliva to the human during its blood meal. Metabolism. Clinical presentation of Lyme disease may include the characteristic bullseye rash and erythema chronicum migrans, as well as myocarditis, cardiomyopathy, arrhythmia, arthritis, arthralgia, meningitis, neuropathies, and facial nerve palsy. B. burgdorferi infections have been found in possible association with primary cutaneous B-cell lymphomas, where a review of the primary literature has, as of 2010, noted that most of the PCBLCs examined have been unresponsive to antibiotics, 846 hence, as in the case of Chlamydophila psittaceae association with ocular adnexal mucosa-associated lymphoid tissue lymphoma, the working conclusion was that if B. burgdorferi is truly associated with PCBCL, then there is wide geographic variability. And other factors are probably involved. 846. Life cycle. Progression of the disease follows from 3 stages. Stage 1 affects the area around the bite, with a swelling or red rash that emerges around a defined center and expands to 15 cm in diameter, 658. Once the rash starts to subside weeks after the first symptoms manifest, symptoms relating to flu may ensue. Essentially, antibiotics are most efficacious to prevent further growth and symptoms of the disease before the primary symptoms manifest. 659. Stage 2 occurs weeks to months after. If left untreated, the bacteria spread through the body and affect the heart, bones, and nervous system. B. burgdorferi may sometimes damage conduction pathways in the heart, leading to irregular heart rhythms, which if significant enough may require the insertion of a pacemaker. Facial paralysis, fatigue, and loss of memory are common emerging neurological problems on the second stage of Lyme disease. If these exacerbate further, the host may develop encephalitis or meningitis. Disease. Stage 3 occurs a couple of months or years after the initial infection, with feasible ensuing developments of arthritis that can affect patients with the disease for a long period of time. Due to the prolonged presence of B. burgdorferi in the body, the immune system activates responses in the joints that rather abets in the worsening of the arthritis. Stage 1. Anaplasmosis and babesiosis are also common tick-borne pathogens that infect humans similarly to Borrelia burgdorferi. Consequently, it is possible for an Ixodes tick to co-infect a host with either two or all other diseases. When a host is co-infected, the combined effects of the diseases act synergistically, often proving to cause worse symptoms than a single infection alone. Co-infected humans tend to display a more severe manifestation of Lyme disease. In addition, they tend to acquire a wider range of secondary symptoms, such as influenza-like symptoms. More studies and research must be done to determine the synergistic effect of CO infection and its effect on the human body. Stage 2. So far, there are three factors that may contribute to the severity of the clinical manifestation of Lyme disease. The presence of ribosomal spacers, plasmids, and the outer surface protein C are indicators of the severity of the infection. Additionally, humans, themselves, vary in their response to the infection. The variation in response leads to different clinical manifestations and different infections to different organs. After the pathogen is transmitted, it will acclimate to the mammalian conditions. Borrelia burgdorferi will change its glycoproteins and proteases on its plasma membrane to facilitate its dissemination throughout the blood. While infecting, B. burgdorferi will express proteins that will interact with endothelial cells, platelets, chondrocytes, and the extracellular matrix. This interaction inhibits proper function of the infected areas, leading to the pathological manifestations of Lyme disease. In response, the host will initiate an inflammatory response to attempt to remove the infection. Stage 3. Borrelia burgdorferi, also, expresses at least 7 plasminogen binding proteins for interference of factor H at the activation level. This is part of a complement system evasion strategy that leads to downstream blocking of immune response. B. burgdorferi was the third microbial genome ever sequenced, following the sequencing of both Haemophilus influenza and Mycoplasma genitalium in 1995. Its linear chromosome contains 910,725 base pairs and 853 genes. The sequencing method used was whole genome shotgun. The sequencing project, published in Nature in 1997 and Molecular Microbiology in 2000, was conducted at the Institute for Genomic Research. Overall, B. burgdorferi's genome oddly consists of 1 megabase chromosome and a variety of circular and linear plasmids ranging in size from 9 to 62 kilobases. The megabase chromosome, unlike many other eubacteria, has no relation to either the bacteria's virulence or to the host-parasite interaction. Some of the plasmids are necessary for the B. burgdorferi life cycle but not for propagation of the bacteria in culture. The genomic variations of Borrelia burgdorferi contribute to varying degrees of infection and dissemination. Each genomic group has varying antigens on its membrane receptor, which are specific to the infection of the host. One such membrane receptor is the surface protein OspC. The OspC surface protein is shown to be a strong indicator of the identification of genomic classification and the degree of dissemination. Varying number of OspC loci are indications and determinants for the variations of Borrelia burgdorferi. The surface protein is also on the forefront of current vaccine research for Lyme disease via Borrelia. Genetically diverse B. burgdorferi strains, as defined by the sequence of OspC, are maintained within the northeastern United States. Balancing selection may act upon OspC or a nearby sequence to maintain the genetic variety of B. burgdorferi. Balancing selection is the process by which multiple versions of a gene are kept within the gene pool at unexpectedly high frequencies. Two major models that control the selection balance of B. burgdorferi is negative frequency-dependent selection and multiple niche polymorphism. These models may explain how B. burgdorferi have diversified, and how selection may have affected the distribution of the B. burgdorferi variants, or the variation of specific traits of the species, in certain environments. In negative frequency-dependent selection, rare and uncommon variants will have a selective advantage over variants that are very common in an environment. For B. burgdorferi, low-frequency variants will be advantageous because potential hosts will not have a built immunological response to the variant's specific OspC outer protein. Variation of severity. Ecological niches are all of the variables in an environment, such as the resources, competitors, and responses, that contribute to the organism's fitness. Multiple niche polymorphism states that diversity is maintained within a population due to the varying amount of possible niches and environments. Therefore, the more various niches, the more likelihood of polymorphism and diversity. For B. burgdorferi, varying vertebrate niches, such deer and mice, can affect the overall balancing selection for variants. Molecular pathogenesis, genetics, evolution, negative frequency dependent selection, multiple niche polymorphism.